Hey everyone, and thank you for joining me here in Boulder, Colorado. Today I'm going to talk about adaptive introgression of the Denisovan variant of the gene MUC19 in American populations. I'd like to take a second to thank all my co-authors, particularly the people of the Huerta Sanchez lab at Brown, and also the people of the Avila Arcos lab at the UNAM. I'm going to talk a lot about archaic introgression. What that means is a process which you can understand as having the ancestor of modern humans and also these other archaic species in which modern humans diversify. And these archaic species, such as Neanderthals and Denisovans, at some point admix with modern human populations. And before they go extinct, they leave some portions of their genome in modern humans, such that some modern humans today are carrying this archaic ancestry. And while it's very small, when you look at a single individual, when you look at multiple individuals, you can actually reconstruct quite a bit of the archaic genome. And so the questions that I like to ask are, how do these archaic genome elements actually affect modern humans, our health, and our lives? And this is important because basically everyone who's alive today carries some amount of archaic ancestry. So this is a map of Neanderthal and Denisovan ancestry in populations around the world. So most people should carry both Neanderthal and Denisovan ancestry. Individuals in Africa actually carry a small amount of Neanderthal ancestry as well as some super archaic ancestry as well. So this is basically everyone who's alive. But once you look at the human genome, the archaic ancestry is not uniformly distributed. There are some deserts of introgression, particularly in genes and in non-genic functional regions that are depleted of archaic ancestry. And this, of course, is the result of very strong negative selection that's been estimated to have removed up to from 10% archaic ancestry to as little as the 2% we see today in as short as 10 human generations. So it's very strong. But at the same time, for some genes, we actually see an excess of archaic ancestry in some populations. And so these are all examples of genes that have been co-opted for adaptation in modern humans. And I want to bring your attention to the gene EPAS1, which is being proposed to help Tibetans survive in low oxygen environments. So this is a haplostrip plot where you can see most Tibetans actually carry the Denisovan version of EPAS1 relative to, say, the Han Chinese. But what I found most fascinating is that when they've estimated the timing of selection, so if this is modern humans and these are the different Denisovans, and there's been introgression 40,000 and 30,000 years ago, positive selection in EPS1 didn't start in earnest until 12,000 years ago, which means it happened uniquely in the human lineage. And that to me has been mind-blowing. This paradigm shift when it comes to archaic ancestry, to realize that these genes don't have to be adaptive in the archaics. The simple fact that they are boosting genetic diversity in modern humans has helped us adapt to novel environments. And so I like to think a lot about Indigenous American populations in this context. So if we look at this little vignette where modern humans carry Neanderthal and Denisovan ancestry, are these colored dots, and modern humans around the world carry different types of archaic ancestry. So 20,000 years ago, the people that live somewhere here in Beringia made the cross into the American continent. Presumably, they were adapted to that subarctic environment, but in a really short amount of time, they actually made it through the entire continent where they found all these novel environments that presumably they're becoming adapted to. And each of them presents unique challenges. For example, they domesticated hundreds of species, they invented agriculture, they developed these complex societies. And so all of these present novel environmental challenges. And it's possible that the archaic ancestry they're carrying is actually helping them adapt to these novel environments. And so thinking about Indigenous American populations in that context, there's this excellent article from Austin Reynolds a couple of years ago where they were looking at the genes with the largest signature of positive selection in North American Indigenous Americans. And so for the list of genes, the genes that has the strongest signature of positive selection is the gene MUC19. What's interesting about this gene is that it's also been identified as having a Denisovan version that is found in high frequencies in populations in the Americas. And so it's possible that what's driving the signature of positive selection at MUC19 is actually the Denisovan version of MUC19. And so the questions that I'd like to ask now are, first of all, is MUC19 introgressed? Did we actually inherit that from Denisovan as opposed to being ancestrally shared by both of them? If it is introgressed, is it enriched in Indigenous American populations? Is it also enriched in ancient indigenous individuals, since I have access to ancient DNA samples? And finally, is this enrichment actually the result of positive selection at MUC19? And so the approach I'm using is similar to haplostrips, in which I am taking the genome coordinates for MUC19, and then I'm looking at the Denisovan version of those coordinates. And I can take both chromosomes and use them to create this reference archaic chromosome, where I can then score against modern human chromosomes, in which genomes that have very few differences or no differences will be considered to have the Denisovan version of MUC19, whereas chromosomes that have large amounts of differences have the modern human version of MUC19. So when I plot them here based on the populations of the 1,000 genomes panel, what I find is, this is distance from the archaic reference right here. So up here means it's the more similar to the modern human haplotype. The ones that are down here are more similar to the Denisovan haplotype. And what I find is that African populations here in green don't actually carry the Denisovan version of MUC19 at all, but Europeans, Southeast Asians, and East Asians all carry the Denisovan version of MUC19 at low frequencies, and then American individuals carry the Denisovan version of MUC19 at somewhat higher frequencies. So in Africa is 0, in Europe is very low, which is consistent with not having a whole lot of Denisovan ancestry. In Southeast Asia, in East Asia, it can be found as high as 12%, but in the Americas, some populations have as much as 30% for the Denisovan version of MUC19. And this is, almost 3 times as high, so it's very interesting. But it's possible that that number could be even higher if you consider that American individuals like myself are admixed. So we have some Native American, some European, and some African ancestry as well. And so there's very good research from 2017 where I can get genome coordinates where they separated those ancestries into those 3 components. And I can use that to actually assess where the Denisovan version of MUC19 is being found. And so once I split them, I found once again that the African proportion doesn't have MUC19. The European proportion has very little MUC19 for the Denisovan version, but then for the Native American segments of ancestry, they have an elevated amount of the Denisovan version of MUC19, somewhat higher than most populations around the world in Colombia, but extremely high in Mexico, Peru, and Puerto Rico. Particularly in Mexico, is almost 50%. So that is very interesting. It's a very compelling story. I wanted to see if I could see the same signature in some ancient individuals, some pre-colony individuals, in which I don't have to separate the ancestries. And so I collected published genomes that have good coverage, which there's actually not that many in the Americas. And then we added 3 novel genomes that we sequenced using funding from the Leakey Foundation. So this is what I found, where I've colored in red individuals which don't really carry the Denisovan ancestry. So I found 74 SNPs that are found exclusively in Denisovans and introgressed Denisovan-like variants. And so individuals in red don't actually carry any of those. For the rest of them, they carry some number of these SNPs because genome coverage is actually very low in some of these genomes. I'm never going to find all 74 of them, but I'm taking the evidence of finding some of them as actually meaning that they carry the Denisovan version of the haplotype. And so here I've plotted the number of Denisovan SNPs versus the genome coverage at this region, just to show you that for some of the ones that carry very few copies of the Denisovan SNP, those are also genes that are very low coverage. So it's some indication that these all actually carry the Denisovan version. It's just that I can't really find all that many of those SNPs. The only exception is the ANSEC1 individual, here Clovis, actually has a single position, which has the Denisovan version, but it has a really high genome coverage. So in reality, this appears to be a recurrent mutation, and that individual doesn't actually carry the Denisovan version of MUC19. But then turning to frequencies again, we can see that for some of these populations, in particular here in Mexico, we find again a really high frequency of the Denisovan version of MUC19. And it's a nice overlap with the ones for Peru and for Mexico, for which I actually have modern and ancient sampling, finding very similar frequencies. In Patagonia, the frequency is very high. Of course, my sampling is very limited. So, but to conclude, yes, we find the Denisovan version of MUC19 in non-African populations, so it's probably introgressed. It's found at much higher frequencies in American populations, including some of these ancient individuals, even though my sampling is very limited. And now, in the future, things I need to do still is I need to figure out if this is actually the result of positive selection. And so I'm doing population branch statistics on these. It's something that I have to do in the future. But also, I need to understand why MUC19 for why the Denisovan migration will be advantageous in American individuals, which is something that I don't quite understand yet. So these are some of my follow-up thoughts for things that I need to do in the future. But with that, I'd like to finish and maybe look at my co-authors and appreciate them again. Thank you.